Okay, good afternoon, everybody. And we're carrying on today our discussion about Darwin and about the development of the early ideas about the descent with modification of species from an earlier ancestor, which was an idea which Darwin conceived to explain these observations that he had made about unity in biology, that species share common features, and yet they're incredibly diverse and superbly adapted to their environments, to doing the things which need to be done for them to survive in their own environments. He wanted to understand that, not in light of the ideas which were prevailing at the time, that things were as they were, that they were created to do that. Rather, he wanted to understand a process which could lead to what he was observing. So we've heard about his journey on the Beagle and how he saw things all over the world. He was able to put together these ideas. But one of the things that was very difficult for him was to develop a mechanism, an explanation of the mechanism of the why. He could see, he firmly believed, for example, that all of the finches on the Galapagos descended from a single common ancestor. But that simple observation is not in itself an explanation of why. Why were those finches that he observed different to their original ancestor? Why doesn't he see the original ancestor there present on the islands? That's the kind of problem that he was trying to answer. And he was trying to answer it for many, many different groups, many different observations. Not only of extant species either. He looked at the fossil record and he wanted to understand, why did things go extinct? Why were things which were obviously so incredibly common eons before, why were they now no longer present, and yet seemingly related things were present? What had happened to drive that process? Now, surprisingly enough, he drew a lot of his ideas from artificial selection. Now, artificial selection is what we observe when human beings modify species deliberately, when we modify species deliberately through our own selective efforts. So, for example, farmers for eons have selected particular crops, have selected particular animals for particular characteristics, and have bred them together to amplify characteristics over and over and over again, so that desirable traits in these crops or in the animals could be amplified. And this has been done for so much and for so long that, in fact, many of these bear very little resemblance to the wild ancestor that they came from. So don't seem able to, there we are. So here's a classic example for you. You may or may not know or realize that many of the brassicas that we eat, the green-leafed vegetables, are in fact all so very closely related. And here are just some of them. These are only a few of them. They're all derived from a wild ancestor, and here it is, called Brassica rapa. It grows wild everywhere, and it's a wild mustard. And in fact, you will see it widely grown in a very similar form as rapeseed, canola and canola. These are all look very similar to the wild ancestor. These beautiful fields of yellow that you see in the spring, the farm fields, canola or rapeseed, these are very closely related to the wild mustard. But agriculturalists have selected these for particular traits. All of these are, in fact, closely related kohlrabi, which is the delicious bulb at the bottom of the stem, broccoli, cabbage, Brussels sprouts, kale, and collards. All of these things are very, very closely related, and they have been deliberately bred. They have been bred by looking at this population of wild mustard and making a single observation. That is, if you look at a population of wild mustard, you'll see within that population a lot of variability. They're not all identical to one another. They inherit different alleles that are spread through the whole population. And you will see some plants, for example, with slightly swollen stems at the bottom. Somebody pulled out a stem, tasted that, and thought, that tastes pretty good. So they selected several wild mustard plants with slightly swollen stems, bred them together, took progeny, bred those together, took the progeny, bred those together over and over and over and over again, selecting always for those with swollen stems, until eventually we ended up with kohlrabi. Some of the plants, instead of having flowers all distributed all along the stem like this, some of them had flowers which clustered together at the top in a bunch. And those were selected over and over and over again to produce broccoli, also cauliflower, these things. These all have flowers which occur in a bunch, and on and on and on. Just select for what look like a desirable trait, breed those that share that trait, breed their progeny that show that trait clearly, breed them over and over and over again, and you select out these desirable traits, and you can do it quite quickly. Now, this is not an evolution by natural selection. This is completely artificial selection. This is selection by human beings for a particular trait that they find desirable. Many of these plants, for example, would do very, very poorly in the wild. And in fact, if you leave crops that show these traits for a long time, they very often revert. Very often, natural selection ensures that the normal genes get expressed, and these artificially selected genes can be eliminated from the population quite quickly. We have to maintain these characters by continually breeding these plants together that show those traits. This fascinated Darwin. Remember, he knew very little he knew almost nothing, in fact, about genetics. But he realized that these genetic inheritances were tremendously important. And he saw in artificial selection, he realized that this was a speeded up version of what he might observe in nature. If there were natural selection pressures which selected for particular traits, then this might provide him with a mechanism for the evolution that he was trying to explain. So this is a simple example. There are many, many crop plants that have been bred from wild ancestors. I'll just mention a few others. For example, wheat, oats, all of these grains, they're all artificially selected from a wild ancestor. Corn, and then on to animal species. Well, we've done it for millions of years. And for example, all of the dog varieties that we see, although these are classified as a separate species, Canis domesticus, they are in fact all derived from the wolf. And they've just been bred for particular characteristics, not only of morphology, as you can see here, but also of temperament. So all of these things which can be inherited, we can select for. these are actually the same. In biological terms, these are identical. In genetic terms, they're virtually identical. All that we've done is select for very superficial traits in these different breeds, to which we find attractive or useful, and they've been bred to actually amplify those traits. Leave all of this group together and let them interbreed over and over and over again without any selection pressure, and they will begin to revert. The natural selection will allow these artificially selected genes to be excluded. So Darwin, he was very interested in all sorts of things. I'll tell you, he also was interested in pigeons. He was fascinated by pigeons. Pigeons have been also another animal that have been bred over and over and over again. And what interested Darwin with pigeons was that they had been artificially selected for traits which were positively deleterious. For example, there's a domestic pigeon called a tumbler, which when it flies, it suddenly stops flying and it falls through the air. That in the wild would make it very, very vulnerable to being predated. What it is, in fact, is that they've been artificially bred to be narcoleptic. They fly and they actually fall asleep in the air, and they tumble, and then they wake and they start flying again. That is a trait which, you know, could not possibly be maintained in a wild population for very long. So that interested him a lot. That artificial selection, you could actually breed deleterious traits. Natural selection is the opposite. Natural selection seems to operate to amplify beneficial traits in a particular environmental setting. So he drew some conclusions from this, and also from some other information I have mentioned before. First of all, he observed very importantly that members of a population, you look at any natural population, you look at it enough, you will see that they're not identical. There's tremendous variability in population. So in our genetic terms, we learned about Mendel, there are many different alleles of particular genes which are expressed in a population. Here's the second observation. This he knew himself, but he looked at the work of a man I've mentioned before, which was Thomas Malthus. And this is the second observation. All species produce far more offspring than the environment can support. And in fact, when you look at animal populations, they tend to remain rather stable as long as the environment doesn't change. They tend to remain rather stable. You have animal populations may boom and bust a little bit, but over time in a particular environment, if the environment remains the same, the environment usually supports about the same number of a particular species. Yet Darwin observed that in breeding, they bred far more than actually survived. Most offspring actually died. He wanted to know why. Why is it? Why do most of the offspring die? And then second of all, which of the offspring die? That he drew some information from Malthus. This is Malthus's model of population and resource. He was actually, Malthus was interested in human populations gathered together, for example, in cities and things like this. And he observed that human population grows exponentially as it does here. That we know. But he stated that unfortunately resources could only be increased geometrically. So here it is, and in a straight line here. And there is a point here which he called the catastrophe, the Malthusian catastrophe, is when the population outstrips food, in his model, food production. But in natural systems, this would be what the environment can produce. So Darwin looked at this and saw that, in fact, as population increases, they begin here to actually compete with one another. Now, we're used to the idea that animal populations compete with one another, with different populations compete with one another. That's interspecific competition. For example, animals are preyed on by other species. Okay, that is a form of competition, which might also contribute to limiting the population size. But what was very important in Darwin's thinking was that the members of the same population, as resources become limiting, members of the same population begin to compete with one another. And when he looked, this was actually a model that he did think about, and that is in beetles, things like this, that produce an enormous number of offspring. He saw that resources, if they became limiting, these animals would begin to compete with one another. They would begin to eat one another's food. They'd begin to depend on the same resources that their progeny and their siblings and everything were also dependent on. But there's another observation to be made here at this point. Look at all the variability that you can see in this picture. These are beetles, they're ladybirds, but they're all recognizably the same species, but they vary tremendously just in their coloration, for example, but they may vary in all sorts of different ways. They may vary in their ability to feed efficiently on a particular food source. They may vary in their response to members of the same species being present. Some may more actively chase members of the same species away. All of these things, all of this variability inside this population, some of them are going to be able to feed more. Some of them are going to be able to exploit the resources. The resources, for example, not only food, the resources may be space, places to live, ability to evade predators. All of these kinds of things, they begin to compete with one another. Some of them do things better than others. And those that do it better are going to have more progeny. They're going to be able to feed more. They're going to be able to make better use of the resources. They're going to reproduce more. And all of those characteristics that conferred benefit on them in this intra-specific competition, INTRA, intra-specific competition between members of the same species, those beneficial traits are going to be handed on to the progeny of those that possess them. And those beneficial traits start to be amplified in the population. Unfortunately, it is the law of nature that some of the population are not going to be able to compete successfully. They're going to have less progeny, and their alleles begin to become rarer and rarer in the population. They may not disappear entirely, but remember that what we are really interested in is the phenotype. We are interested in the actual expression of genetic information. That is what is driving these processes. What we see when we look at all the variability here, we are looking at different phenotypes, all in the same species, but it appears as this tremendous variability. Now, that is even more important in those organisms which produce, first of all, reproduce very rapidly, and second of all, which produce enormous numbers of progeny, such as things like the bacteria, the fungi, those sorts of things. And we'll refer back to this in a while. These processes, in fact, take place more quickly in those organisms which have a rapid turnover, which have a rapid generation time. These processes will operate more quickly in those kind of populations. So individuals with inherited traits, which are going to help them survive, those traits are also going to help them reproduce better than others, and they tend to produce more offspring. We have a particular way of referring to this. We say that these individuals are fitter, F-I-T-T-E-R, they are fitter than others. Fitness does not refer to their muscular strength or whatever. Fitness refers specifically to their reproductive ability. They are fitter because they produce more offspring with their beneficial traits than other members of the same species. So this difference in fitness now leads to the accumulation in the population of the beneficial traits. The beneficial traits those with beneficial traits begin to take over the population and crowd out those that don't have the beneficial traits. But realize that the process is not a finite process. It's an ongoing process. As those organisms which are fitter here begin to dominate the population, they begin to compete with one another. Again, those which are fitter than their fellows, they will again reproduce more. And in this way, we slowly begin to see the entire population changing. And what we're watching is evolution in action. So the things which actually eliminate organisms from a population are interesting in themselves. And we need to realize that there are many, many, many different things which operate on an organism in nature, which affect competition, which result in organisms competing with one another in particular. Food resources is only one, but organisms must also compete for space. They must compete for mates. They must, some of them will maybe better than others at evading predation. So they need to be adaptable to environmental conditions. Some of them are going to be less tolerant of certain physical conditions than others. All of these things serve as natural selection on the population. We're just going to see that as populations go through time in an environment, the beneficial traits will become more predominant. The less beneficial traits gradually get lost from the phenotypes that we observe. So that is natural selection. But you can understand how Darwin had been able to draw so much information from artificial selection. It's just that in natural selection, the environment itself, whatever is the component of the environment, whether it's where they live, what they eat, what eats them, what they have to do to get a mate, etc., all of the physical conditions, these are all selecting for particular traits, and those favorable traits will become the most common within the population. The other thing is that the competition between members of the same species is so intense that even the slightest advantage is significant and may allow a particular allele to be perpetuated where others are not. At the same time, the organisms are becoming more and more suited to their particular environment with all of its constraints, whether it's food, space, predators, disease, whatever. the whole population is adapting. The other thing to realize is that if that environment changes for any reason, if a new predator comes in, if a new disease comes in, if the climate changes, if any of these things happen, a whole new set of selection pressures is going to be exerted on the population. And we may see that population change rapidly. Do that over millions and millions of years, and we begin to understand how it is that we see the kind of diversity and the kind of specialization that is present in biological systems. So this is basically just a summary of what I've said, but it produced this natural selection has operated over many, many millions, hundreds of millions of years. And it produced, remember that even the slightest advantage is significant. And that is how we get the evolution of these extraordinary features. For instance, here is a moth there, perfectly camouflaged against dry leaves behind it by looking like a dry leaf. How? Why? Because anybody who looked slightly like a dry leaf in this environment was camouflaged. Even if the original ancestor only very slightly looked like a leaf, progeny who looked more like a leaf would survive longer. The predators couldn't see them. They would breed more frequently. They would breed better, and their progeny inherited the traits which made them look like leaves. These leaf-like moths compete with one another. They compete intensely with one another, compete for space, they compete for mates, they compete for all sorts of, anybody who has a slight advantage does better. When that advantage is to look like a leaf, this leaf-like character becomes amplified and amplified and amplified. Here you can hardly even see this moth here. Looks just like a bump on the bark. It's perfectly camouflaged. Same idea. It's the same idea. Anybody who looks slightly like the bark, they're going to have an advantage. Those moths bearing that allele are going to be at an advantage over and over and over again. Those who look less like the bark get picked off by predators. Those who look more like the bark, they are being selected for breeding with one another and amplifying the allele within the population. They look more and more and more like a stem and the bark of the tree or shrub that they are on. Now, here's something that's very important to realize, that evolution does not operate on an individual. When we talk about evolution, we're talking about populations. We're talking about whole populations. Natural selection increases or decreases heritable traits in a population, not in an individual. An individual is only one of probably hundreds of thousands or billions in that population, in that species. And what surrounds that population that operates on it. It's the environment that population operates in, which is going to increase or decrease the frequency of alleles and heritable traits in a particular population. When we talk about population evolution, which is our next section, you'll understand this a bit better. It's very important to realize that these pressures operate. They certainly an individual is picked off by a predator, and its genes and its alleles are lost from the population forever, while others are left alone in the population and their alleles persist. But if that allele only sticks with the one individual, it's not going to participate. We have to think about these alleles as being almost in a pool, which is the population itself. And that is what natural selection rather operates on. Okay, so that was Darwin's basic theorizing about how evolution took place. It took place adaptation, descent with adaptation, but that adaptation driven by natural selection of traits which made certain parts of the population fitter than others. So first of all, most of what he decided and formulated was based on direct observation. This is such an overwhelming concept for trying to explain how all of the living things that we see around us at the moment have arrived where they are. That it's impossible to perform a single experiment or something which now proves, yes, well, that applies to everybody. But the theory, the hypothesis that he put forward was supported, first of all, by many, many instances of direct observation, such as his observations on the Galapagos, where he looked at the finches on the Galapagos, he looked at the finches on the mainland and made a connection between the two. The Galapagos Islands were very young, the mainland was very old. The species which he saw on the Galapagos had arisen from a mainland group of finches which had arrived on the island after they had formed, and natural selection had driven the evolution of many different populations on the islands, each with their particular traits that made them highly suited to particular environments on the island. So direct observation was certainly extremely important. But remember that what Darwin was theorizing was that we were observing descent with modification. And the similarity between organisms is a reflection of their common ancestry. Even though they may look very different at the moment, when we look carefully at them, we see that very often they share features. There are shared features which reflect their common ancestry. And the more different they are to one another, the more tenuous that similarity is, we can infer that those have been separated for a long time. Those which remain very similar to one another, they separated less in less time. So this was the second thing. We look at those similarities, they can tell us they can give us a lot of information, and that we refer to as homology. So we'll see some examples of that. The next thing, which was tremendously important for this whole thesis, is the fossil record. Because the fossil record, if we look at it really carefully, tells us a tremendous amount about what preceded what we see now. And it fitted the theory of descent with modification absolutely perfectly. And if descent with modification takes place because of natural selection, then the fossil record can show us a tremendous amount about what the kinds of selective pressures that were operating during the evolution of a species, as we'll see in a minute. The last is something which has been amplified in more recent times. It fascinated Darwin, but he, as I told you before in our last class, he had no real explanation for it. And this is the similarity of species which are widely dispersed, widely separated by geography. And this is a reflection of the fact that the surface of the Earth is not static. It is changing. The land masses actually change their conformation over time. One time, a landmass may be a single landmass. There may be a single population of organisms there. That landmass splits apart into different pieces. Those move apart from one another. and the different populations are subjected then to different environmental conditions as they move across the surface of the earth. The populations on them evolve as natural selection drives descent with modification differently on the two. But we see similarities. They still reflect their common ancestry, even though they may be very different to one another. This is referred to as biogeography, and biogeography needs to be considered in conjunction with all of the geological information we have about how the continents have changed over time. So we'll have a look at all of these. Direct observation. Well, we have seen already ample evidence from Darwin's studies, but also from many others. And although I'm not going to talk about it here, I always like to mention him because he tends to be forgotten. This is Alfred Wallace. While Darwin traveled the world, and he especially used South America as one of his prime examples of populations evolving by descent with modification. Alfred Wallace worked in Indonesia, which is often thought of as being like an evolutionary hotspot for many reasons, a lot of them geological. It's a volcanically very rich area. There's been a lot of rapid geological change over the eons, and this has driven very rapid speciation in many of the groups in Indonesia. Alfred Wallace worked in that area and developed exactly the same ideas as Darwin, descent with modification by a process of natural selection. And it's really those two huge bodies of direct observation that developed the whole thesis of evolution, of mechanisms for evolution that we know now. But we can also see it very easily if we do some simple experiments. And remember what I told you about for example, those species which have a rapid turnover, which have a short generation time. The modifications in the populations under changing conditions can take place very, very rapidly, fast enough for us to observe them. Most evolutionary processes take place over such a very long time that the only way we can really observe them is to go back into the fossil record in one way or another. But in some species, we can actually see it happening right within our, easily within our generation time. So there are many examples of this. But this is a classic one discussed in your book if you want to read more about it. And it depends on observations on this bug. Bug, by the way, is a proper zoological term. A bug is an insect with a sharp beak that they insert into plants or insert into whatever they feed on. This is a soapberry bug, and what it does is it shoves its long beak down through a fruit, actually into the seed. It doesn't feed on the fruit itself, on the flesh of the fruit. It feeds on the seed. And this is important. It has a natural host, which is a balloon vine. So this is in Southern California. And this is if you measure the beak length, then you find that on the natural plant that it lives on, it actually has a short beak length. It doesn't have to burrow very far into this fruit to find the seed and feed on it. However, the soapberry bug will actually feed on other things. This is its normal food source, but it will feed very usefully. It will feed on introduced species. Now, this golden rain tree in central Florida is something of a pest, and soapberry bugs are actually encouraged to feed on it as a way to to try and limit it. And when you look at populations of soapberry bugs on these introduced golden rain trees, what you find is the seed lies much further below the surface of the fruit than in the balloon vine. The golden rain tree was introduced into Florida about 40 years ago now. This is 35 years, I think, but it was about 40 years ago. And when you look at populations of soapberry bugs on these, the soapberry bugs that feed on golden rain trees have a longer beak. They have a longer beak. How did that happen? Well, in a population, in a normal natural population, have a look here. Have a look here, and you'll see here's the beak length. Here's the population distribution for beak length. Here's the mean. Yeah, the median rather there. And so in a normal natural population, some of them have longer beak lengths, some of them have shorter beak lengths, right? But transport them to the golden rain tree, and you find that the number of bugs with a different beak length. I think I made a mistake actually, that these have shorter beak length, right? Yes, I'm sorry. Natural is longer. Sorry, in the introduced it is shorter. Sorry, that's a bad mistake. If I knew how to edit these videos, I would edit that out so that I didn't look like an idiot. But in any case, the point to be made, have a look here and you'll see that there's a wide variability in the natural population. But move them to a new environment, move them to a new food source, and the population changes. The population now, the beak length actually, average beak length actually decreases. This to such an extent that, in fact, these soapberry bugs have been introduced to areas of the world where this golden rain tree is a pest, such as Australia. And if you go to those populations, you'll find that they have very similar beak lengths to these. So this is an example of very rapid change, but look where it is. Do we look at a single individual soapberry bug and say, oh, well, this has evolved? No. What we look at is the whole population. We've still got variability, but the mean is much lower in this environment than it is here. Natural selection operates on a population. Even though individuals may carry or be at risk, we view the whole process as taking place on a population, not on an individual. So here's another classic example, and this has important implications for us in our modern life. So if you want to talk about rapid generation times, there's nothing faster really than the prokaryotes, the bacteria. And this is a classic experiment with bacteria looking at their resistance to antibiotics. So if you, I'll just describe for you here, perhaps make sure that you label the diagram that I put. What I've shown here, this is a petri dish here, and what I've done is I've spread bacteria all over it, just some ordinary bacterium like Staphylococcus aureus or something like that. And on the petri dish here, I placed an antibiotic disc. Never mind what the antibiotic is, but this disc here, a bit of filter paper soaked with antibiotic, and that antibiotic diffuses into the agar. And what we observe after several days, letting them grow, this is thickly grown, completely overgrown with bacteria, but around the antibiotic disc, there is a clear zone. And in this zone, bacteria, these bacteria can't grow because the concentration of antibiotic is too high. And very, very often when you do this experiment, you will observe that inside this zone, which we call the zone of inhibition, you may have a few colonies of bacteria which are actually resistant, which actually grow. If I was to subsample these and grow them in an antibiotic-rich environment, I'd find that I could select 4 bacteria which were resistant to this antibiotic. Here's what's really important about that. Seems simple enough, okay? What I'm doing is I'm in a way, this is kind of artificial selection, right? I'm selecting for a particular trait which is resistant to this antibiotic, and I'm amplifying it. Well, there's some really important implications from this. And it is to ask this, I need to ask this question. Did this antibiotic cause these to be resistant? The answer is no. The antibiotic did not make them resistant. The selection does not cause a mutation to occur. What the selection does is it selects those individuals which already have the mutation. The mutation has to be present. That allele has to already be present. The selection pressure is not going to force an allele to suddenly appear. The allele has to be present, even if it's extremely rare. Even if it's extremely rare, it has to be present in order to be selected for. This is tremendously important because it's very easy to believe, to get confused and feel that natural selection is forcing alleles to occur. It does not. Natural selection operates on already existing alleles. In this case here, if we looked throughout this whole population, we would find the occasional bacterium which was resistant to this antibiotic. The only place that we can visualize that is in the environment where that pressure is being exerted in the area of this antibiotic. And here, a few of our bacteria were able to grow. They have been selected for. Now, this is the second implication of this. Has little to do with our discussion about evolution, but very important in human medicine and in clinical medicine, because whenever we use antibiotics, we run the risk of selecting for those bacteria which are already resistant. And if antibiotics are not administered very carefully, if they're used carelessly, if they're used too frequently, or if they're used in too low a dose, we run the risk of selecting for resistant bacteria. And this indeed has happened over and over and over again in the last 80 years or so, 70 years that we've had antibiotics widely available in the human population. We have found over and over again that bacterial infections, which used to be very easy to eliminate with antibiotics, are no longer easy to eliminate because we have forced the development of whole populations which are resistant to. The most serious one at the moment is methicillin-resistant Staphylococcus aureus. Staphylococcus aureus is probably the commonest cause of infections such as skin infections, infections of surgical wounds, all of those sorts of things. And for a long time, Staph aureus was very, very easily dealt with penicillin. Penicillin was very widely used. Penicillin was used even for mild infections. It was used for throat infections. It was used for this, for that, for the next thing. But all of the time, we were selecting for those Staphylococcus aureus which were resistant. Take somebody who has an infection, give them penicillin, and what are you going to do? You're going to kill off the penicillin sensitive bacteria, and you run the risk of amplifying those which are resistant. And that indeed is what has happened. We've run the gamut of antibiotics all the way up. Each time this happens, we develop resistance, we try to use a new antibiotic, a new, more powerful antibiotic. And Staphylococcus aureus has run now the gamut of virtually all of our antibiotics, right up to this antibiotic called methicillin, which is related to penicillin, but is a very, very powerful, more modern antibiotic. And now whole populations of Staphylococcus aureus are found widely in hospitals all over the world, which are methicillin resistant. And they are referred to as MSRA, methicillin resistant Staphylococcus aureus. It's a very dangerous use of antibiotics like this can be extremely dangerous. And what you're watching is evolution in action. The antibiotic is selecting for an allele which is already exists in the population, but is often rare, but becomes amplified because of the selection pressure produced by the antibiotic itself. Okay, so those are the kinds of direct observations that we can make and that have been made of kind of evolution in action. Here's another, and this is to look at homology. And homology is a reflection of common ancestry. And it's a very, very powerful evidence for evolution and for descent with modification. It depends on the fact that related species have characteristics with an underlying similarity, even if the actual function is completely different. And this depends initially in biology, this depended on careful anatomical examination of homologous structure. That is, we looked at things like limb bones and that sort of thing across a wide range of animals and saw how they were similar and how they were different. In modern times, this kind of homology can be extended to nucleic acids. We can look, for example, at the sequence of DNA, and we can actually see homology, and we can track through comparison of DNA structure, we can track ancestries. So but it is basically using the same theories, the same technique, the same theories with a different technique. Let's have a look, for example, at limb bones, and in many vertebrates, these share an amazingly similar structure, even where function is completely different. Now, these here are all limb bones of mammals, and here showing completely different functional use, but incredible similarities of structure. Here's our human. This is our hand, and the humerus, radius, ulna, carpals, metacarpals, and the phalanges. First of all, to notice, this is a pentadactyl limb. That is, it has 5 digits. And that pattern is preserved pretty much through all of the mammals. Have a look here. This is a cat. This is a cat leg, but look what happens. The cat actually, where we have a flat hand and a flat foot that we walk on, the cat actually walks on its toes. So its metacarpals are extended up as the wrist is, so the wrist is vertical. It basically walks, that's its palm. For us, this is our palm. The cat, that's its palm there. and the thumb is much reduced. Nonetheless, there's just huge, tremendous homology. Now look at something radically different. Look at the whale, and we see the same humerus, here radius and ulna, carpals, metacarpals, and phalanges, but now adapted for swimming. Look at the bat, also a mammal. It's also humerus, radius ulna, metacarpals, carpals, phalanges. The phalanges extended and skin in between to form a wing so that it can fly. But we could extend these homologies. First of all, it needs to be said, these are all reflections of the fact that the mammals all share a common ancestor. There's a common ancestor to all of these, an ultimate common ancestor from which they were all derived. The second thing to be said is we could extend our study of homology. We could be looking, for example, at reptiles, at lizards and things like that, and see that they also have much the same pattern. It gets a little bit complicated when you look at things like the snakes, which have secondarily lost their limbs, but we'll mention them again in a minute. All of these homologies are reflections of common ancestry. There was a common ancestor. When you look at morphology like this, we can say very often that the more different the homologies, the more different these organisms are from one another, the further back the common ancestor lay. But we can't put a time on it. We can only say this happened before this, this happened before this. But when we look at molecular homology, when we look at DNA sequence, when we follow the evolution of individual genes, we can in fact put something of a clock on it, because we know how often, we know more or less how often mutations occur in DNA. They occur with a certain regularity over time because they're random. Mutations are random events, and they act like a clock. When we look at particular DNA sequence, we see how much mutation has accumulated between different DNA sequences. We can say, within bounds, we can say how fast molecular change has actually occurred. So that's really important, modern development. Okay, here's a second thing, which is really fascinating, and that is, if we look at the embryos of related organisms, we can see that they share structures, they share homologies, which completely disappear in the adults, but reveal very, very deep ancestry. So, for example, here in vertebrates, all vertebrates, there are a number of, these are only two of them, there are a number of different features which all vertebrates share. But here are two really important ones. Here is a chick embryo, here is a human embryo. The same thing, by the way, can be said of you could take virtually any vertebrate embryo and put it in here, and you'd be able to see the same things. That they have what are referred to as pharyngeal arches there. You can't see it clearly, but you can see more clearly actually on the human. Here, the arches are actually supported by cartilage. They do disappear. They end up being incorporated, for instance, in the cartilages of the throat, that sort of thing. But in the embryo, these are actual slits. There are actual slits here, and they also all share a post-anal tail, even though it may be reduced. Here it is here. In birds and humans, it's very much reduced. In other vertebrates, of course, their post-anal tail is much longer, but we all have one. We all may even have vestigial but we all have that. When we look at this, it reflects actually a very, very deep ancestry, way beyond the vertebrate ancestry in actual fact. And it reflects an ancestry which we will, if you stick around for the second part of biology in the next semester, we'll talk more about. But this very definitely reflects a common ancestor to both of these. And if we were to go to any vertebrate embryo, we would find the same thing. We all share a common ancestor. So as I've mentioned, molecular homologies are also really, really important. And one of the interesting things about molecular homologies is we can sometimes find, for example, that in related species, a gene may have been completely silenced, never ever used, where it's very important in another related species. And we only know that because we can sequence the DNA and we can see the similarities. There is nothing in the phenotype of the silenced one to tell us that the gene is there, but it is. And that is, again, a reflection of common ancestry. Similarity of genes, even if they're not expressed, is a reflection of common ancestry. So using these similarities and trying to somehow place in time the ancestry, whether it's distant or recent, etc. The one way we can do that is by drawing up what is referred to as an evolutionary tree. And evolutionary trees attempt to explain hypotheses about the relationships between groups. And this is very often done by homology. It used to only be done by morphological and homologies, like the similarities of limb structure and the degree of difference between limb structures. In evolutionary trees, we determine the degree of relationship, the time of relationship, if you like, by the most recent common ancestor to a particular group. I'll show you, there's something that's a little bit confusing about evolutionary trees, but I'll show you when we actually look at one now. It used to only be done for morphology. They carefully tried to build trees that reflected evolutionary history of particular groups. Say, for example, the evolution of the primates. They would try and link, they'd be able to link the monkeys together, the New World monkeys, the Old World monkeys, the great apes, etc., and try look at the morphology and try and say, well, they shared a common ancestor here, they shared a common ancestor there. Nowadays, these evolutionary trees are very much amplified by our studies of the DNA and of DNA sequence. And in fact, they very often, fortunately, they very often correlate very well with one another, and the information, the two sets of information, morphology and molecular information, can be put together to develop really finely detailed trees. So let's have a look what we're going to look at as we go through a tree like this. We're going to look at shared characteristics, and shared characteristics in a particular group of organisms reflects the fact that that group has a common ancestor. So here's just one thing I just want to explain really carefully about these evolutionary trees. Because this mammals here are sitting next to the amphibians here, does not show their degree of relatedness. What you need to look at is, where is the common ancestor? So if we look here, let's just take the mammals since they're so familiar to us. Here's the common ancestor here to all of these other vertebrates here. Okay, so the mammals are more closely related to all of these than they are to the amphibians, because they share a more recent common ancestor. You can think of it like this. This is like one of those mobiles that you hang, right? I can flip this over. I could flip it over like this and have the mammals at the bottom. And the true relationship is still there, because this is what you must be looking at. Not what it's sitting next to, but where the common ancestor is. So first of all, let's have a look at this entire group, because it's such an important group for us. So first of all, invertebrate terms, as we look through all of the vertebrates, our nearest, most distant vertebrate relative. I know that sounds strange, but you'll understand what I mean. Our nearest, most distant relative are the lungfishes. And because all of the tetrapods, all of those organisms which have 4 limbs, basically have 4 limbs. That includes the snakes, by the way, because they derive from the lizards. All of these have 4 limbs. We all descended from a group of fish, and currently the closest living relative are the lungfishes. Here they are here. You can just see they have peculiarly structured fins, which became available for natural selection to operate on over geologic time, evolutionary time, to produce the 4 limbs of the tetrapods that we see here. So the shared characteristic here for all of these is that they all basically have limbs with digits, and we have 4 limbs with digits, unless there's some spectacular particular modification. So that's the amphibians, mammals, lizards, snakes, crocodiles, ostriches, and then the birds. So you can see all of them have these 4 limbs: 1, 2, the wings 2, 1, 2, 2 wings, 1, 2, 3, 4. Snakes are specialized lizards in actual fact. And some snakes, in fact, have a pelvis and rudimentary limbs as well. It's a special loss of the limbs. There is a specialization, but that group, in fact, are also tetrapods. Mammals, of course, that's us. We have 4 limbs. Hello, and if you could see my feet, then you'd realize that I do indeed have 4 limbs. So all of these share that characteristic. It is a reflection of the fact that we share a common ancestor. This must have developed, the 4 limbs with digits must have developed beforehand, because we all have it. Okay, we share the common ancestor. This characteristic here, a little bit difficult to explain if we haven't talked about embryology or anything, but this is a particular feature of embryo development. It's a structure around the embryo called the amnion. It's a protective membrane. And in actual fact, this protective membrane is what allowed the vertebrates to penetrate onto land. It appears in two forms. The one is in an egg, and the other is in live birth. And we all share it. The mammals, lizards, snakes, crocodiles, ostriches, and hawks. You'll immediately say, oh, wait a minute, surely these sit out separately. Yes, they do. Mammals are separate, but they're not separate because of the amnion. What you need to remember is something important here, and that is that in each of these groups, there are those who lay eggs. Even in the mammals, there are those which lay eggs. Even in mammals which have live birth, they still have an amnion, even though they don't lay eggs, but there are those that lay eggs as well. So live birth, for example, does not pull us out of this grouping. Instead, we are grouped because all of these have an amnion. We share a common ancestor with all of these, okay? The next homology to look at is taken. There are others as we go through, but and the next one we'll look at is the possession of feathers, and that currently links all of the birds. But if we go into the fossil record, of course, we also find that there are dinosaurs, for example, which have feathers as well. These now look at the relationships. Are the birds here? They clearly share a common ancestor, but they look here, and you'll see that they share a relatively recent common ancestor in the crocodiles. So that this group, the birds, are closely related to the crocodiles. They are also, and that group together, crocodiles and birds, are closely related to the lizards and the snakes. They are, in fact, believe it or not, all reptiles. Many, many taxonomists regard birds as being specialized reptiles. They certainly share a common ancestor. They much more recently share a common ancestor with the reptiles than they do with the mammals. However, this tree also tells us that this whole group, the mammals, lizards, snakes, crocodiles, ostriches, the birds rather, we all share a common ancestor. We all share a common ancestor, and in fact, that ancestor was probably reptilian. We diverged after the common ancestry, but the common ancestor was very probably a reptile. These groups here have preserved the reptilian characteristics much more closely than we have, but we are all related by a common ancestor. There's a huge amount of information, a huge amount of evolutionary information that we can derive from a tree like this. And what is really important, this tree is built up simply from morphological and some embryological information. This tree is actually very closely supported when we look at molecular information as well. And it all goes to the point that this huge diversity of organisms here has all arisen from descent from a common ancestor, even down here somewhere, a common ancestor. Descent with modification has produced all of these. The time of that, the time that diversification took place is not always clear, but we can glean an idea. We know, for example, that the amnion, development of this characteristic, took place long before the development of this characteristic here, of the feathers. The development, the diversification into those which had possessed an amnion took place before the diversification of those with feathers. Now, with molecular information, we can, in fact, begin to try to put real dates to these. But for this kind of tree, all you can say is this took place after that, which in itself is useful, because we can also go now, we can use a tree like this and now go to the fossil record and try to see what does the fossil record tell us about this kind of descent. Okay, I just, to finish off today, I'm just going to mention one thing which is a little bit confounding on all of this. And that is that we can see very similar morphological and functional features in different species, which are not in fact homologous. They have arisen from entirely different evolutionary sources to the same purpose. For example, flight. Here's a classic example. This here is a sugar glider. Sugar gliders are actually marsupials, and they are in Australia. Marsupials are a primitive group of mammals. They're one of the most primitive groups of mammals. And here they are in Australia. Australia was isolated very early on, so it has a really interesting and often bizarre fauna, closely related to the earlier mammal faunas. Sugar glider, it has a skin which stretches between its front and back legs, and it literally glides through the air. And here in North America, we have flying squirrels. Some of you might even have had flying squirrels as pets. They do exactly the same thing. They have their skin stretched between the front and back legs, jump out of the trees, and they are able to glide. And they're actually able, both sugar gliders and flying squirrels are actually able to guide themselves and get from one place to another by gliding. They do exactly the same thing. They're totally unrelated, except for the fact that they are both mammals. This feature has arisen completely independently of one another, because it suited these organisms better to their particular environment. The selective drive was the same, but in totally different animals. So there's no homology here between the two. This slight characteristic does not relate them to one another at all. And so we say that it is analogous. It is, and this is an example of what we refer to as convergent evolution. Most evolution is divergent, leads to the development of new species, these different groups. This is convergent evolution. They look the same, but they're actually unrelated to one another. You need to be very careful because in biology, because sometimes these convergent features can actually confuse the issues tremendously. Okay, I'm going to leave it there for today, and then we'll carry on with this on Monday.